Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the deep dive. This week, we're digging into sources you sent about, a really tough challenge, finding better Alzheimer's treatments. But the twist here isn't some brand new, super complex drug. It's actually something derived from cinnamon. We're looking at a follow-up analysis of a trial on sodium benzoate. Yeah, you heard that right. It's a common food preservative, but chemically speaking, it's what your body makes after you eat cinnamon. What if, just maybe, slowing down Alzheimer's could involve something you can just take as a pill? Okay, let's unpack this.
That accessibility factor is precisely why this line of research is so potentially impactful. We know the fundamental issue in Alzheimer's is this buildup of harmful proteins, mainly those amyloid-beta peptides, often called A-beta. Now, the treatments recently proved, they're scientific breakthroughs, no doubt, they actually target and remove these A-beta proteins, slowing down the disease. But the logistics are tough, they're very expensive, require regular IV infusions, think hours in the clinic, and they carry some pretty serious risks. We're talking potential brain swelling or even bleeding.
Right, that's a major consideration. You've got these powerful but high-risk, high-cost options versus this huge need for something, safer, effective, and frankly, just easier for people to take at home. And that search for something simpler, safer, it brought researchers back to sodium benzoate. Can you just quickly clarify for us, what does metabolite of cinnamon actually mean here?
Sure. So when you eat cinnamon, your body processes its compounds, like cinnamaldehyde. A metabolite is just what's produced during that breakdown process. Sodium benzoate is one of those end products. And crucially, it's already approved as a food additive, deemed safe. That gives it a massive leg up in terms of developing it as a potential therapy. And there was already promising background research, right? Animal studies showed this stuff. Take it orally could reduce that amyloid protein load and actually improve memory and learning.
And earlier human trials backed up the safety part. People with mild Alzheimer's taking daily doses, 750 or 1,000 milligrams, showed improvements in their cognitive scores. You mentioned the safety was like placebo, basically no significant side effects at those doses. That sounds almost too good to be true. Are there any minor concerns at all, maybe digestive stuff, since it's a preservative?
That's a really good point to raise. While the overall trial showed safety similar to placebo, which is excellent, it is a food additive.
Yeah.
Very high doses theoretically could cause some mild stomach upset in some individuals. But honestly, when you compare that potential mild issue to the risk of brain edema from the IV therapies, it's a completely different ballpark. The safety profile here looks incredibly favorable. An oral pill versus serious infusion risks, it's night and day.
Okay, that context is vital. So the original trial showed safety and some cognitive benefit. This current study we're diving into by Lin and Lane, it was a secondary analysis asking more specific questions, right, about how it might be working.
Exactly. They had two key questions. First, could taking sodium benzoate orally actually lower those A-beta peptide levels circulating in the blood? Show a direct biological effect. And second, was there a connection between how much A-beta a patient started with and how well they responded cognitively? Basically, could we predict who benefits most?
Right, trying to find the ideal candidate for the treatment. So they dug back into the data from that original trial. Remind us of the setup.
It was a solid design. 24 weeks, randomized, double-blind, placebo-controlled. Involved about 149 patients in Taiwan, aged 50 to 100, all with probable mild Alzheimer's. They were split into 4 groups. Placebo or daily sodium benzoate at 500, 750, or 1,000 milligrams.
And the researchers measured cognitive function using that ADAS-Cog scale and the actual A-beta protein levels in blood plasma. Let's pause on the ADAS-Cog scale for a sec. How should we think about that score? What's a good outcome?
Okay, so with ADAS-Cog, a lower score means better cognitive function. In Alzheimer's trials, just slowing down the increase, the decline, is often seen as a win. But if a patient's score actually decreases, that means they're potentially regaining some lost ground cognitively. That's a really significant finding if you see it.
Got it. Lower is better. So with that setup, what did they find regarding the protein levels in the blood? Here's where it gets really interesting.
Well, they combined the data from the groups that showed the cognitive benefit before the 750 and 1,000 milligram doses. And compared to placebo, taking those effective doses led to a significant reduction in one specific peptide, a beta 140. And also a significant reduction in the total sum of the measured A-beta peptides in the blood.
But not the 500 milligram dose.
No, the 500 milligram dose didn't show a significant change compared to placebo. It seems there's a threshold effect.
Okay, so higher doses lowered the proteins, but the second finding, that's the one that really connects the dots, isn't it? The predictive link.
Yes, this is the really compelling part. They looked specifically at the patients taking those effective doses, the 750 or 1,000 milligrams. And they found that the patients who started the trial with higher levels of a different peptide, a beta-142, in their blood, those were the ones who showed the greatest cognitive improvement over the 24 weeks. Their ADAS-Cog scores dropped the most.
Okay, hang on. Why the switch? First you said a beta-140 was reduced, but now a beta-142 is the predictor. Why is 142 so important here?
Good catch. These A-beta peptides aren't all the same. A beta-142 is generally considered the more problematic and stickier form. It's the one that's more prone to aggregating, clumping together to form those amyloid plaques that are characteristic of Alzheimer's.
So finding that the treatment works best in people who start with more of this stickier, plaque-forming protein, that really suggests it's hitting the core disease process, right?
Exactly. It implies the treatment is most effective when there's more of the key pathological target present. If you have more of the bad stuff, you stand to benefit more. It strongly suggests a targeted mechanism.
And what's fascinating here is this connection was only seen in the groups actually getting the sodium benzoate, right? Not the placebo group.
Precisely. That specificity is key. It wasn't just random variation. It was clearly linked to the treatment itself interacting with the patient's specific level of this key protein.
And the sources suggest this might not be just about reducing a beta. There's talk of it being a multi-target therapy.
Yes, that's another layer that makes this compound interesting. What's fascinating here is, while reducing a beta fits the dominant amyloid hypothesis, other research pointed out by the sources shows sodium benzoate also seems to boost the body's own natural antioxidant defenses, specifically things like catalase and glutathione.
Antioxidants. Okay, how does that link into Alzheimer's? I thought it was all about the plaques.
Well, the plaques are central, but they cause downstream problems. All that protein buildup creates enormous oxidative stress in the brain cells. Think of it like cellular rust, damage caused by unstable molecules called free radicals. Catalase and glutathione are like the body's internal rust removers. They fight that oxidative stress. So if sodium benzoate boosts these natural defenders, it could be helping protect the neurons from that secondary damage while also potentially reducing the primary A-beta driver.
So it could be hitting the problem from two different angles, reducing the protein buildup and helping the brain cells cope with the stress.
That seems to be the implication, supporting both the amyloid idea and the oxidative stress theory of the disease. It broadens the potential mechanism quite a bit.
That definitely adds to the appeal. But as always, we need to look at the caveats. The limitations, what did the researchers themselves flag as important unknowns?
Right, critical reflection is essential. This raises an important question. The trial was only 24 weeks long. That's relatively short in the context of a chronic disease like Alzheimer's. We saw good effects over that period, but we don't know if they last long-term. Does the benefit plateau? Do you need to take it continuously? Those are open questions.
And the study population was quite specific too, wasn't it? Han Taiwanese participants.
Yes. While the basic biology should apply across populations, you always need studies in more diverse ethnic groups to confirm the findings are generalizable before you can think about widespread use. Standard practice.
But maybe the biggest question mark, the one that really gives pause, is the blood versus brain issue, right? They measured protein changes in the blood.
Exactly. That's the elephant in the room, scientifically speaking. We know sodium benzoate reduced A-beta peptides in the peripheral blood plasma.
But Alzheimer's is a disease of the brain. We always hear about the blood-brain barrier being this, selective gatekeeper. How do we know if lowering a beta in the blood actually translates to lowering it in the brain where the damage is happening?
That is the crucial gap. Changes in blood A-beta often correlate with changes in the brain and cerebrospinal fluid, but it's not always a direct one-to-one relationship. This study shows the drug gets into the blood, lowers proteins there, and that links to cognitive improvement. But it doesn't definitively prove it's clearing plaques inside the brain itself. We're inferring that link based on the cognitive outcome, but we don't have the direct proof from this specific analysis.
So the obvious next step for research isn't really about safety or dose anymore. It's about getting a look inside the brain.
Absolutely. Future studies must incorporate techniques like PE scans, brain imaging, that can actually visualize amyloid plaques. They need to directly show that taking oral sodium benzoate reduces the amyloid load in the brain.
Without that visual confirmation, it remains a very promising candidate, but not yet a proven brain-acting therapy.
Precisely. Direct evidence of target engagement in the brain is the non-negotiable next hurdle.
Okay, so let's try and synthesize this. Pulling it all together. what's the big picture here? So what does this all mean? We've got compelling evidence that sodium benzoate, this simple, oral, safe compound that our bodies make from cinnamon, appears to lower key Alzheimer's-related proteins in the bloodstream. And importantly, that reduction, particularly for patients starting with higher levels of the stickier beta 142, seems linked to real, measurable cognitive improvements in mild Alzheimer's.
Yes, the research really strengthens the case for sodium benzoate as a potential novel beta-reducing therapy. And the safety and oral administration are huge advantages over current options. And the fact that this potential therapy comes from a metabolite of a common spice, something many of us consume regularly without a second thought, it's a powerful reminder, isn't it?
It really makes you wonder what other common compounds, maybe hiding in plain sight in our diets, might have untapped therapeutic benefits. If something derived from cinnamon could potentially tackle Alzheimer's through both amyloid and antioxidant pathways.
It encourages a broader perspective, perhaps looking more closely at the complex chemistry already present in nature and our food, not just focusing solely on inventing entirely new molecules.
A fascinating thought. Keep looking for those unexpected links between what we eat and how our brains function. There might be more hidden gems out there.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.